Question 3, that one is your plant one. So what is it that it's giving you? It's really beautifully describing to you an investigation into gases that are being tweaked and changed by a plant. So you've got a setup with a light source, a cold water in a clear pipe situation, air at a standard temperature. The plant chamber required at a standard temperature. They're stressing the temperature bit a couple of times here. Why are they repeating that? Hint, hint. And then your carbon dioxide measuring device with air being pulled out from that plant. So it gives you the mass of carbon dioxide absorbed by 10 tomato plants measured in 1 hour was measured at so and so at 40 degrees. The same group of plants were grown at the same light intensity at all temperatures. And now the first question is actually just into why is there cold water in a pipe just under the light source? Lamps do heat up. You still want the light to come through, hence a clear pipe. Some of you have misinterpreted that clear pipe as it's refracting the light or changing the frequency, you're turning it into some sort of color. It's not really a triangular prism. You would have used a color filter if you wanted to create a particular wavelength. So blue filter or a red filter, a green filter, and see what happens then. But this is just a clear pipe just to allow the light to get through, but to stop the temperature from being affected by those light bulbs. And why is it important? Why the same 10 plants were used? So the same 10 plants were used. Why do they need to be the same? Let's say some of them have got far more leaves than another plant. That's a higher surface area, more photosynthesis. Let's say those plants were grown, some on really great soil with all the minerals available, and the other ones had magnesium deficiency. They will not have functional chlorophyll, low numbers of chloroplasts. So individual plants have got individual characteristics, and their rate of photosynthesis would be different. The mark scheme so far, the clear pipe with cold water is to prevent the increase in temperature, and the different plants would have a different mass or surface area or different number of chloroplasts or anything else that would have an effect on the photosynthetic rate. The question on the control, careful, the question above, that was your control variables. This one here is a question on the control experiment. Whenever you see this question, and it will come up in different contexts, if it's a control experiment, think of that one as the placebo kind of equivalent, running the experiment with absolutely nothing relevant in it to see if actually just the basic setup would have an effect. In this context, that means run this experiment again, but just don't put a plant in it. Don't put anything in it at all. A bit like a placebo pill, which has, I don't know, starch, and that is it, to see whether there is actually an effect of just taking a pill. Yeah, I'll make a point of that every time we're doing practicals from now. So repeat the apparatus, and so same conditions, everything the same, just no plant. This question here, I think many of you did the clever people mistake that you start reading the question, but you don't read it until the end and your brain is already in overdrive. You're answering something amazing, just not to do with this question. So go slow and actually read it. Describe how the experiment could be modified to determine the mass of CO2 released by respiration. You've got a plant. A plant does respiration. It does respiration day and night because it's alive day and night. So in the light and in the dark, it will do respiration. But if you want to actually just get to the respiration CO2 producing rate, that's what you want to do is you want to get rid of the photosynthetic production, sorry, the photosynthetic absorption of carbon dioxide. So turn the light off. Do the whole thing again, just without light. That's the key thing here. To get to the respiration rate, do the whole thing, but in the dark. You then have what clearly looks like a maths question. It asks you a little, hinting you in the right idea direction, kind of moving you in the right path question on this. So you have got given the temperature of the chamber. It wants from you how much carbon dioxide was absorbed. It should maybe say in total, but maybe that would have been giving it away a bit too much. You have got carbon dioxide produced, and here in this last column, it doesn't say carbon dioxide absorbed. but rate of photosynthesis, that literally is how much carbon dioxide has been taken up by the plant. So your overall here, have a look at what the question actually asks you to do. Explain why both the mass of carbon dioxide absorbed during photosynthesis and the carbon dioxide produced. Why do you need the absorbed and the produced to get to the overall rate of photosynthesis? So why would you need this number and this number to get to that number? The idea here is that that's your total. And that's the missing thought process that is trying to hint you to get to. So your 30.4, take away your 0.2 to get to that number. Literally take the rate of photosynthesis number, take away the respiration number, because it's literally how much carbon dioxide is there absorbed, how much is there made, and then what's overall absorbed. Your numbers, if you want to pause the video on the numbers for a second, you do that. So the question that was just above is the true rate of photosynthesis given by the total mass of CO2 used, which is the mass absorbed, as well as the mass produced by respiration. So expressing that in some version or another, various ways of wording this would have been absolutely fine. And then comes your long one. That's a 4 marker here. And it speaks to you beautifully about the sweetness of the sugar content of tomatoes, and it varies with temperature. It doesn't automatically say the hotter it gets, the higher the sugar content, because that's not exactly true. And some of you have written just that the hotter it gets, the more sugar there will be. Look at your information here, the rate of photosynthesis. Here we go. Just that last column here. Where do you have your highest rate of photosynthesis? Your 35.3. So it doesn't seem to be that 40 degrees is the ideal temperature for whatever overall enzyme reaction is going to be. Maybe for the individual enzymes, 40 degrees as temperature would be optimum. However, for the overall process, it isn't. Okay, and several of you have gone into the direction of just talking about enzymes and go, okay, 40 is best, but actually, no, it isn't. 40 seems to be best here. If you look at this number, 40 seems to be the best temperature here. It seems to be an increase here in what is actually wasting the sugar, wasting the carbon that you could have put into sugar in respiration. So the plant is obviously doing some processes to keep its temperature steady overall. We will do the route transport and everything else, so that the explanation for that is still to come. But that number here should have been identified. That number here should have been identified as the highest. So there's an increase in the rate of photosynthesis until temperature 30. However, then there's a decrease. Why is that happening? Because you've got an overall increase in these numbers here. Sorry, I'm talking in terms of the numbers. The mark scheme itself has said photosynthesis increases from 15 till 30, which means more sugar is produced. However, above the 30, less sugar is produced. Why is that? Respiration just seems to increase and increase until the 40 degrees. We don't know above that. At some point, the plant will die, but we don't know above that. Which means that some of that sugar is broken down in some other processes as the temperature increases, which means that if you were to increase the temperature further, there's not actually a benefit in terms of more sugar being produced. So if you were a farmer and you want to produce super sweet tomatoes, having too hot of a temperature is not actually going to produce you those amazingly sweet tomatoes. And some sort of reference to data, I can give you a mark there as well. It then gives you something about wind speeds and stomata. Now, some of that is component 3, but some of that is a little bit of common sense as well. So an advantage and a disadvantage. If the stomata, and that is a concept from GCSEs as well. So yeah, I've been nice when marking, but I didn't just let you get away with nothing there. If the stomata open, why is it an advantage? You're going to have the gas exchange, and you will need to use the word diffusion into the leaf so that your carbon dioxide can diffuse into the leaf, the oxygen can diffuse out of the leaf, not just movement. I need to have it slightly more specific. You're A-level students, so as much language as you can put in there that shows you as capable with that. And then the disadvantage, if the stomata are open, you're also going to lose water through them. You may want that as a plant, you may want the transpiration stream, but that is component 3. So yes, you should really get an extra mark or 2 here, but then again, it would not give you a status quo right now. So advantage is less water loss if they're closing, disadvantage carbon dioxide cannot diffuse in mouths if they're closing, or the other way around. Then it describes the change to you. The production of chemicals by enzymes contribute to sweetness. When the tomatoes are kept at cold, the genes for those enzymes that actually lay down the sweetness in the tomatoes, those genes are being switched off. They're being methylated. So if the plant is too cold, the genes that code for the enzyme that then makes the sweetness, those genes are being switched off. It tells you it's a permanent change in gene expression for that plant while that plant is alive, but it doesn't speak about germline or not germline at this point, so we don't know any of that yet, but we do know that methylation is a way of switching off that is epigenetic. So that is an epigenetic one that was done beautifully. However, that bit here wasn't quite done beautifully. So use the information given and your knowledge of protein synthesis. Explain how why the sweetness of these tomatoes will not increase after they've been stored at low temperatures, even if afterwards the temperatures are higher. So this is part of this. You've got 2 marks. Part of this is your own knowledge. What do you know about protein synthesis? Protein synthesis starts with the DNA. You read the DNA, you make your messenger RNA, you splice it. You've got your exons, they become your active messenger RNA, goes to the ribosome. Amino acids attached together to make your protein. In this case, that's your enzyme. So the enzyme, that's the pathway, your knowledge of protein synthesis. Where in this whole process is that process interrupted? Right at the start, if the DNA is methylated, you can therefore not read it. The enzymes that do the mRNA polymerase and other enzymes cannot access that gene. You can't start reading the gene. Therefore, you can't make the enzyme. That enzyme is therefore then no longer there to make the sugars. And that won't change even if you afterwards change from the low temperatures. They told you here it's a permanent change. So it does not undo itself. The mark scheme says that messenger RNA is not produced or no transcription. Therefore, you can't have translation taking place. Therefore, no enzyme will be produced overall because the DNA was methylated. Right, that was question 3.